Trachea, histology of trachea. The trachea is a flexible tube lined with a highly specialized respiratory wall. Its structure is primarily organized into four distinct histological layers: mucosa, submucosa, fibromuscular cartilaginous layer, and adventitia. Mucosa, innermost layer. Epithelium, pseudostratified ciliated columnar epithelium. It contains ciliated cells, propel mucus and trapped particles upward to the pharynx. Goblet cells secrete mucus to trap inhaled debris. Basal cells act as stem cells to regenerate the epithelium. Lamina propria, a layer of loose connective tissue rich in elastic fibers that separates the mucosa from the submucosa. Submucosa, composed of loose connective tissue containing numerous mixed seromucous glands. These glands produce secretions that humidify the air and trap smaller particles. Fibromuscular cartilaginous for fibromusculose cartilaginous layer, cartilage, supported by 16 to 20 C-shaped rings of hyaline cartilage. These rings keep the airway open and prevent it from collapsing during inhalation. Brachialis muscle. The open ends of the C-shaped rings face posteriorly directly against the esophagus and are bridged by a bundle of smooth muscle known as the brachialis muscle. Adventitia, outermost layer, made up of loose connective tissue that anchors the trachea to the surrounding structures in the neck and mediastinum. Contains blood vessels, nerves, and adipose fat cells. Some more slides of trachea. Next, epiglottis is the epiglottis is a flexible leaf-shaped flap that prevents food from entering the trachea during swallowing. Its core is made up of elastic cartilage that is sandwiched between two distinct mucosal surfaces, adapting to both digestive and respiratory functions. The epiglottis has three main histological layers. Elastic cartilage. The central core of the epiglottis consists of elastic cartilage. Chondrocytes reside in lacunae, and the matrix is rich in branching elastic fibers, visible with special stains like Werkhoff's or Elastica van Giesen, giving the structure the flexibility required to bend over the larynx. Anterior lingual surface. The surface facing the base of the tongue is covered by stratified squamous non-keratinized epithelium. This is continuous with the lining of the pharynx and contains numerous taste buds. Posterior laryngeal surface. The surface facing the airway changes as it goes down. The upper half is lined with stratified squamous non-keratinized epithelium, which transitions into pseudostratified ciliated columnar epithelium with goblet cells in the lower half. Lamina propria and submucous. A layer of connective tissue lies beneath both epithelia. The lamina propria on the lingual side is relatively loose, whereas it is dense on the laryngeal side. This layer contains diffuse lymphatic tissue, nerve fibers, and numerous seromucous glands that secrete moisture into the airways and trap contaminants. Epiglottis. Next, lungs alveoli is down. The lung is divided into two primary zones: the conducting zone, airways, and the respiratory zone, where gas exchange occurs. The entire organ is enclosed by the pleura, a double-layered serous membrane. Conducting zone. The conducting zone is responsible for warming, humidifying, and filtering the air. Trachea and bronchi. Lined with pseudostratified ciliated columnar epithelium, which contains goblet cells to secrete mucus. The walls feature cartilage rings and submucosal glands for structural support, and moisture. Bronchioles. Yeah. As airways narrow, cartilage and goblet cells disappear. Epithelium transitions to simple ciliated columnar and then simple cuboidal epithelium. These structures contain club cells, formerly Clara cells, which detoxify harmful substances and secrete surfactant-like material. Smooth muscle is prominent here, adjusting airway diameter. Respiratory zone. The zone includes respiratory bronchioles, alveolar ducts, and alveoli, where oxygen and carbon dioxide diffuse. Alveoli, microscopic thin-walled air sacs that provide an immense surface area for gas exchange. Their walls consist of three primary cell types. Type 1 pneumocytes, flat squamous epithelial cells covering approximately 95% of the alveolar surface. They form the ultra-thin barrier necessary for rapid gas diffusion. Type 2 pneumocytes, cuboidal cells that produce, store, and secrete pulmonary surfactant, a fluid that lowers surface tension to prevent alveolar collapse. Alveolar macrophages or dust cells, phagocytic immune cells that patrol the alveolar surface, engulfing inhaled dust, debris, and bacteria. Interstitium and blood vessels. Interstitium, the thin connective tissue space sandwiched between alveolar walls and capillaries, contains elastic fibers to support lung expansion and collagen. Blood-air barrier, where type 1 pneumocytes meet the endothelial cells of the pulmonary capillaries. This ultra-thin membrane allows oxygen and carbon dioxide to exchange passively. Pleura, the outer surface of the lungs and the inner chest wall are covered by the pleura, which consists of a single layer of flat mesothelial cells supported by connective tissue and elastin. Lung alveoli under microscope. The pupils. So today I discuss the respiratory system, the histology of the organs of respiratory system. Thank you.